Astagfirullahaladzim. Cek.
Iya.
Assalamualaikum warahmatullahi wabarakatuh. Good morning and welcome everyone to today's seminar. The Honorable Dr. Rika Harini, SSE MP, as Head of Environmental Geography Department, and respectable all participants of this seminar. First of all, let's thank the Almighty Allah Subhanahu wa Ta'ala who has given us some mercies and blessings so that we can attend this agenda without any troubles cycle. Secondly, salawat and salam may always be given to our Prophet Muhammad Sallallahu Alaihi Wasallam, who has guided us from the darkness to the lightness. Before we go to the main agenda, I would like to introduce myself. I'm Najwa Nurawalia from Environmental Geography Studies, and I will be your master ceremony this morning. It gives me great pleasure, and I'm excited to be hosting it. On behalf of Geography faculty, I would like to extend a warm welcome to all of you, especially our speaker, for taking the time to attend this event. We have an exciting panel of speakers lined up for you today, and we hope that you will find their presentation and discussion informative and engaging. So sit back, relax, and enjoy the seminar. Today's seminar will talk about high throughput sequencing or HTS technologies to provide environmental microbiome with Professor Annie Hu from Institute of Urban Environment, Chinese Academy of Science. We have distinguished speakers who will be sharing their insights and knowledge on this topic. I also want to give a reminder that there will be probably an assignment or task, especially for block students, and the tutorial lecture will inform the details later on. So I urge all of you to sit back, relax, and absorb the information that will be shared today. It promises to be an insightful and enriching experience for all of us, and I hope that by the end of the seminar, we'll have a better understanding of the HTS technology and its importance in our lives. Next agenda, we would like to, there will be a welcoming speech. So allow me to invite Dr. Rika Harini, SSE MP, as the head of Environmental Geography Department, to deliver her welcome remarks. Please welcome Dr. Rika Harini. The time is yours.
Thank you, Mbak Naswa.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to thank to Allah Subhanahu wa Ta'ala. Secondly, I would like on behalf on the Environmental Geography Department to the Environmental Hydrology and Climatology Laboratory for the currently holding this webinar. So we know because the webinar is one of the program of the Environmental Geography Department. So in the Environmental Hydrology and Climatology Laboratory is a second monthly webinar in 2023. So in this morning, I would like thank for a speaker, Professor Dr. Niyu. This is from Institute of Urban Environment, Chinese Academy of Science. Good morning, welcome from this webinar. So I'm very apologies because I can join this webinar because after that I will have class. So I hopefully this webinar can be useful to increase our knowledge and can make a solution related the environmental issues and problem. So once again, I would thank from the committee, especially from HKL laboratory and all you participants, especially in a block student. So I hope this webinar provide additional knowledge and regarding from environmental issues. So be enjoy and successful and Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Waalaikumsalam. Thank you so much, Dr. Rika, for your delightful speech. And so without any further ado, I'd like to invite Mrs. Dr. Lintang Nurfadila, MSc, as our moderator. Time is yours.
Thank you, Mbak Najwa. Good morning, everyone. So our speakers already joined us today. Hi, Prof. An Yihu. Good morning, Prof.
Yeah, good morning.
Good morning. Thank you for joining us today. So as I mentioned before, most of our participants are from environmental geography student in 7th semester, and I hope that our biology student and also animal science student will join us also. So actually, this kind of topic is still new for us. So I hope, but during my lecture in water quality and lake hydrology, I always give them insight about what is antibiotic resistance. But during this lecture, I hope that it will provide us a new insight into the antibiotic resistance. So before you will start your lecture, let me introduce you as our speaker today. So our speaker today is Professor An Yihu, PhD. He's a professor at the Institute of Urban Environment, Chinese Academy of Science, and his expertise includes environmental DNA, microbiological biography, microbiological source tracking, antimicrobial resistance, and disrupting effect of emerging contaminants. And he specializes in investigating the response mechanism of microbial biodiversity and functions to anthropogenic disturbance in urban water system. So you will have about an hour for your lecture. So
Okay.
the time is yours. You can start it now.
Okay, thank you. Okay, thank you for introduce and thank you for the invitation. And it's my pleasure to be here and to introduce our research progress. And yeah, today I would like to give a presentation about how to use the high support sequencing technology for fire environment technology. I'm An Yihu from Institute of Urban Environment, Chinese Academy of Science. So first I will introduce a little bit about our institute, and then I will introduce my profile again, and then we will go. I will go to the main topic of today's presentation. So first, I would like to show the location of our institute. Actually, IUE is located in Xiamen, in southeast China, and actually it's not far from University of Jakarta. And I think maybe we'll have a chance to visit Jakarta in September. Hope to see you again. And here is our institute. Our institute is located in Xiamen, and was launched from 2016. So it's long history. Actually, we are a new institute in Chinese Academy of Science. So our institutes have different departments and focus on urban environment issues. We have a department focus on urban environment and healthy, and also have department to do the pollution control, and also we have a central and focus on ecology environment, and we also try to develop some technology to do the urban waste recycling and material recycling, and to make our city better. And so basically we have three research subject area. So first is environment effect of urbanization, because we know now our countries and our cities and become more and more and peoples, and the city become big and big. So we need to to know their environment effect of urbanization, and also we know the residential of cities and produce waste day by day. So we also need to find a way to do the pollution control, and also to need to develop some new technology to help us to recycling the waste. And because we have limited resources, and we also want to we also have an area to focus on urban development strategy and planning management, because we need to find a better way to build our cities. So because in most part of our institute need to do experiment, and so we need to have a well equipment instrument center. So yes, we have instrument center, and the total value of instrument center is more than 1 billion RMB, and we have 24 specialist technicians, and are responsible to management of this instrument. And we also, our institute, participate in the national evaluation to see whether our instrument center and maintain good or not. And you can see here, and in last several years, our instrument center and did great work, and in 2021, they score as excellent in national evaluations. So who are and we are environment bacteriology lab, and we have several staff, and me, and also Professor Chang Bingyu. Right now, Professor Chang Bingyu is working as a professor in Taiwan University, but he also is adjunct professor of IOE, and also we have associate professors Jiang Wei Li, and we also have several assistant professors. And we also would like to introduce Dr. Bob Adia, and he graduated this summer, and to continue to stay in our lab as assistant professor. We also have like Chinese students and international students, and because you can see here, actually, our lab is international. It's a quite international lab, and the main research focus of our lab is we want to address the environment microbiology issues, and also trying to use some technology from environment engineering and environment chemistry, and to do the multidisciplinary research. And in the last several years, and we have successful several international students successful get PhD, a degree from UCAS, and you can see here, and we have students and from Africa and from Bangladesh and from Iran and also from Indonesia. So they did great work, and actually Dr. Bob Adoya and Dr. Edward have got excellent student certification from UCAS in the last several years. So for me, actually, I'm a microbiology ecologist. So as introduced by the host, and I focus on environment DNA. So for this part, we can assess the biodiversity of microorganisms. But right now, we also can use this technology to assessment microorganisms like fish and other kind of animals. We also can use microbial to do some source track analysis to identify the potential pollution source. And also, right now we know a lot of synthetic chemicals used by people and used for livestock or discharged into the surface water. So right now the effect of emerging contaminants become more important and receive more and more attention. So I also have a part work to do the disrupting effect of emerging contaminants. And finally, I also interested in antibiotic-resistant genes because they are critical for human health. So actually today I will introduce some work related to the environment DNA and the microbial source and the disrupting effect of emerging contaminants. So okay, let's move on the main topic of today's presentation. So first, the question is why should study environment microbiology and how to study environment microbiology using cutting-edge technologies. So we can see here, the picture shows a tree of life. This is a phylogenetic tree of life. We can see here the bacteria here, archaea here, the microorganism, and the eukarya. Human also belong to eukarya. So you can basically see from this tree the bacteria, archaea, the microorganism contribute a major part of genetic. So that means, in other words, they have a lot of genetic resources, and also they have critical research and research meaning for us to do the evolution. So we need to know how human can evolve from the cell to now, right? And also microorganism maintains the ecological functions, but right now only 1% of them can be cultured. So we don't know who are they. We don't know what are they doing. So the conventional method approach just trying to isolate, but is quite less efficient. So right now we need to use molecular technologies to identify the microorganism to know what are they doing. So what are they doing? Actually, microorganism drives the global biochemistry cycling. So we can see from these three figures. So first figure shows the carbon cycling, and second figure shows the nitrogen cycling, and the plot C shows the surface cycling. So you can see here, for example, for carbon cycling, microorganisms can not only fix carbon dioxide, but they also can respirate like humans. We breathe, so they also can produce carbon dioxide. So if we want to get to to balancing the carbon in future and control the climate change, we need to care of the microorganism, because we want to to make them to fix more carbon dioxide, but to produce
Hi, Prof.
uh,
Yeah, I think we lost for a moment. Hi, Prof. Can you hear us? Can you hear my voice? We can see your presentation, but we cannot hear your voice. Yeah, Professor Anihu will quit and then join us with us again. Okay, we will wait. Hi, Prof. Hi, Prof. Yeah, we can see your presentation. Would you mind to try to check your sound? Yeah, yeah, we can hear you now. Yeah, we can hear you now, but it's a bit, I think it's a distortion with the voice. Can you please try to check the voice again? Check the microphone. Sorry, we have a little bit trouble with the sound, but Prof. Anihu is now changing his laptop, so we could wait for him. Please do not hurry. Are you with us, Prof. Anihu? Maybe you can stop screen, and then yeah, maybe you can stop screen first, and then yeah, try to. Or should we close this Zoom and rejoin? Go ahead.
Okay. Yeah.
Yeah, we can hear your voice.
You can hear me now?
Yes,
Okay.
finally.
Yeah.
Sorry about that. Yeah,
Yeah,
we
sorry.
know that.
Okay.
Yeah,
So, okay, let's move
yeah,
on.
please.
So sorry for that, and I don't know why, because the voice is work in my laptop, but I don't know why. Okay.
Yeah, it's okay, Prof. Can we start now, or there's trouble again?
Hello.
Yeah,
Okay.
please. Yeah, please start.
Sorry.
Again, Prof.
I think it's due to internet, but I also bring Bob here like as a backup. Sorry for that.
Okay, sorry about that, but it's okay. It's fine.
Okay. So yeah, I'm just saying the microorganisms actually is the important for Earth because they maintain the ecosystem function, and actually they also are critical for human and animal healthy. From this figure, we can see now actually in every place of our body, like our skin, mouth, gut, and everywhere the microorganisms are there. So actually, the United States and other countries, I mean, they did a lot of work related to human microbiome to understand why microorganisms are there and what are they doing. Right now, we know that actually microorganism is very important and is a main part of our bodies. And if we weight their biomass, we can see that microorganisms actually account for more than 30% of our weight. So if we want to have a good healthy, actually we need to have the balanced microorganism in our body. So if we have a good microorganism in our bodies, they can good for our healthy and also can promote the fast growth of animals. If for this is very important for living broad and also can enhance the production of agriculture. So also can enhance the efficiency of fertilization. So this is what the balanced microorganism can do. But if we introduce a lot of chemical contaminated pollutions into into the soil, water, and we eat a lot of artificial or like industry food. Actually, it's not good for our body. Will cause the imbalance of microorganism, and the basic patterns from those imbalance microorganisms. We can see the decreased diversity, and also we can see the huge change in microorganism microbial community composition. And for that, actually, will cause some bad healthy. For example, if we get fevers, and for sometimes we will get the bacterial infection. That's actually is caused by the imbalance of microorganisms in our bodies. So another issue actually is related to the antibiotic resistance. So we know that if we get a fever, if we get infection, we need to take antibiotic and to kill the bacteria, kill the pathogens actually. So the antibiotic have we have different type of antibiotics. They have a different mechanism to kill the bacteria. But in other hand, the bacteria also have their own biological mechanism to resist the antibiotic to keep them survive under the antibiotic pressures. So if we, give more antibiotic, actually the bacteria can resist it, they still can survive. If the pathogens and get this kind of ability, then we will have a big problem for our health. So this data is from the British government, and in 2014 they released a report that antimicrobial resistant infections actually will become as a top problem to cause the death, and in 2015, and it will exceed the cancers related deaths. They also predict in 2015 which area will become a serious area to face the antimicrobial resistant infections. We can see here the Africa, South American, and the Asians, and we may face the serious problem to deal with the antibiotic resistance. Is actually is critical for our healthy. And so we need to know the basic questions. So we need to know who are they and what are they doing. What can they do for us? Yeah, finally we want microorganisms can help us to improve our healthy, to help us to control the climate change, help us to make the city better, right? So we need to use a different method to answer this question. First is cultural based. We need to isolate the bacteria in lab and then identify what are they doing. But use this less efficiency method, and even right now there is some kind of high throughput isolate approach. So right now the molecular technology become popular and become more popular. So they can use some biomark to identify the bacteria composition, and also can use metagenome and to know the microorganism community function. And also they also can use the molecular technology to identify the virus, because we know actually for COVID-19 it is originated from the environment, right? So we need to pay more attention for environmental wearables. We need to know what kind of wearables they are and whether they can cause a problem in the future. And we also can use mass transcriptomic and to know whether they are live, because in the environment microorganism we can't see the microorganism, so we don't know whether they are live. They are active to play some functions. So we need to identify their identifying them at mRNA level to know what are they doing. So the voice is okay now? Okay.
Yes, Prof. Sorry, I still muted.
Okay, sorry. Okay, so why use the high throughput sequencing technology? And for this figure, we can see this is a species distribution and the curves for environment. So for you, this the horizontal axis show the species number, and the vertical axis shows the abundance of each species. So we can see here in environment, actually just a few, maybe dozens or hundreds species account for more than like 80 or 90 biomass of the total community. That means just a few species can be identified by using the conventional method, because they have limited detection threshold. So we need to use the high throughput sequencing. So we can detect, for example, the conventional method can detect those species, but the high throughput sequencing can detect most of them. Why they are important? Because, for example, like pathogens, actually in environment they count a small part. They belong to realm biosphere. So if we don't use this kind of technology, we can't detect them, right? So this figure just shows the typical flow of how to do the environment microbiome. So first we do sampling, and then in lab we will do the DNA RNA extraction, and we do the sample preparation, and we loading them into sequencing platform. But the most important is bioinformatics. We need, because we will get big data, we need to know how to analyze the data to get the pattern we want to know. So as this figure just shows the typical the data analysis flow of the biomark based sequencing or metagenome sequencing. So what kind of data we can have? So first we can know their diversity, and we know that for biodiversity conservation, we need to assess the environment organisms, their diversity, and we also can know whether they have different community composition. For example, if we analyze the healthy human, or a human have disease, the microbiome in healthy body, or the human have this disease, we definitely will see they have different micro community, because the healthy human will have a balanced microorganism. And we also can identify what kind of microorganisms they have. And we also can use some kind of stochastic analysis to identify which species or which taxa and cause such difference and such difference. And we also can, at some time, we can measure different kind of environment parameters. In water, we can measure temperature, pH, dissolved oxygen, and in our body, we can measure like blood pressures and also other kind of metabolic parameters. So we can identify which parameter cause such change in microbial organisms. And we also can use the network to know and whether such kind of microorganisms can work together and cause to maintain the community functions, and also can identify their phenotype, use phenotype tree to identify the potential evolution pathway. And in the following parts, I will use some research cases and our research cases to give some example. First, I would like to show how to identify the pollution signals based on the microbiome analysis. Actually, in the following parts, the main research are from river. So here is Xiamen, actually is located here, and our institute is located in Xiamen. In a nearby area of Xiamen, there is a river named as Jiulong River, and it consists of two tributaries, North River and West River. Why this river is important? Because actually it's a second largest river in our province and provide important resources for drinking water and agriculture and industry activities. But this river is polluted, have the serious pollution levels. And for North River and the upstream of this area, actually is have a lot of livestock, live breeding activities, and a lot of swine there. And in West River, this is an agricultural area. So in these rivers, they have problems for nutrient pollutions. For North River, livestock breeding and release a lot of ammonia in that area. But in West River, due to agriculture, due to fertilization, we found a high concentration of nitrate. So we want to know whether such pollutions will cause a problem and cause the change in microbial community composition of in those rivers. So we go to the field and collect the water and sediment samples, and after we identify the microorganism microbial community compositions, we can put this data in the figures. So from this figure, we can clearly see that, okay, the water sediments have a different microbial community compositions. This is because they have different environment conditions. And also we can see here. So we can see here, actually in water, we can see here is a dark color represent methanogens. And actually methanogens usually they live in sediments because they don't like oxygen. But in surface water, they are full of oxygen. So why we see a lot of anaerobic microorganisms there in surface water? This is because actually due to the pollution and due to the livestock and due to the wastewater, and they may link or flow into the river, into the surface water, then cause the introduce the microbial pollution. Actually, why? I mean, we also can see they have a different pattern in two rivers. Why? This is because the nutrient concentration, and they have different conditions in nutrient and in nutrient species, right? So actually, the environment and cause the problem, and also the human activities also cause the microbial pollutions. So how can we identify such problems? So basically, we can see here from this figure, we can see here actually microorganisms are everywhere. For example, our bodies is also a kind of environment for a microorganism, but they have a different species in certain environment. In other words, our bodies, like human animals or the water or soil, they have different kind of microbial species. So we can use this characterize to identify whether the human-related or animal-related species enter into the water. If the human-related species are there, so we can know, yes, okay, this water is contaminated by human activities, by human or animal waste. So we use these methods to identify the potential pollution source of microorganisms and also antibiotic resistant genes, because we want to identify the pollution sources, and then maybe we can write a proposal to government to let them to control the pollution source to migrate the water pollution in our area, right? So first we use metagenome to identify the abundance of antibiotic-resistant genes in our rivers. This is North River, this is West River. So basically, we can see here, this is a line here. This is a line shows that this is a level 3. That means in Jurong River, the antibiotic resistance may cause some pollution because they are above this level. So that means we didn't control the antibiotic resistance very well in this area. So where are they from? We use a microbial source track to identify. They may come from the sewage, they may come from the animal feces, or from the natural sources. So we can see here in the whole area, most of antibiotic resistance in most sites are originated from the sewage. That means we didn't, maybe in this area, the wastewater treatment plants didn't maintain well, or maybe they need more wastewater treatment plants to deal with the sewage. And we also can see here, maybe you can remember that, as I said, in upstream area of North River, there are living stock breeding. Yes, we see here animal feces cause problem in upstream of North River, especially for the dry seasons, because dry season have a low water flow, so may enhance the proportion of pollutions. And then we also identify the microbial species to see whether those species from the human gut or animal gut may appear in this area, because we already see sewage and animal feces are here, right? Based on the antibiotic-resistant genes analysis. Yes, we also see the high proportion of the gut-related or sewage-related microorganisms visualized. That means we use a different approach to solidify our findings that, okay, in our area we need to have a better management for the living breed and for waste to deal with sewage. And okay, then we also investigate the problem caused for the nitrate pollution, because we know nitrate pollutions and it's not good for the what they may cause the algae bloom, right? So we need to identify the nutrient pollutions. So in this work, we use the nitrate dual isotopes because they can identify the potential pollution source of nitrate, whether they from the fertilization, whether they from the sewage, or from the soil. But the problem is this method can't distinguish manual and sewage pollution. In this area, they have wastewater treatment plants. They also have living stock breeding. So we want to identify whether the manual sewage cause such problem. So we incorporate the microbial source track to identify to help the isotope to distinguish the pollution from manual and sewage. So we collect samples from not only from the river but also from the soil and the swine wastewater and the influent effluent of local wastewater treatment plants. In this figure, we can show here, okay, the different environment. Yes, they have different microbial communities. So this is this is the foundation of microbial source track. Different environment have different microbial community composition. So we use this kind of these environments as a source and to track their contribution to the surface water of like main channel of river, tributary of river, and reservoir. So from here we can see, okay, the influent and effluent of wastewater treatment plants contribute a large part of microorganisms of the river waters. But for swine wastewater, they contribute little. So from this figure, we can put their pollution source in the map, and we can see here, okay, in the cities, the main pollution from the sewage. So that means we still need to enhance the management of wastewater treatment plants, but for the living stock breeding, right now it's okay. Yeah, we also to put the isotope parameters and the microbial source track result together to see whether they give the similar findings. Yes, we can see here. So this is the afferent for microbial source track. This is isotope signals, and the efferent micro isotope signals. They provide similar findings, and in other words, if we combine the different approach from different research area, we enhance our understanding of the water pollution problems. Okay, and after we know who and the distribution of microorganisms, and we know that they can be used to track the pollution source of water, right? So we also want to know whether the local microorganism in rivers can change, due to the pollutions or due to other kind of environmental changes. So how to evaluate the responses of microbiome to environmental changes? So here I would give the research cases about emerging organic contaminations. So as I said, the antibiotic and the chemical we use like sunscreen and the material used the plastic bag or plastic bottles, and they belong to emerging organic contaminations. And we use them daily, and finally they will enter into wastewater treatment plants. But right now, the wastewater treatment plants can't deal with those kind of contaminants very well because they have very low concentrations, and they also are resistant to the current treatment technology. So finally, those kind of contaminants will enter into the aquatic ecosystem, will cause a lot of problem for the environment. So actually, yeah, based on previous report, more than 90% of consumed prescription drugs finally will end up in our wastewater, where we are entering into the surface water. So the typical effect of emerging contaminants cause the problem for fish. And for this we can see that actually some kind of compounds can cause the fish get wrong genders. That means will cause the decrease in male fish and increase in female fish, and finally will cause the serious problem for our environment and ecosystem. So we need to identify their disrupting effect, right? But the problem is actually we use too many kind of chemicals. So the one problem is we're complex, and the second, in the local environment, there are other kind of stresses like temperature, oxygen, nutrients, but we still need to do because they will cause some less effect. That's, in other words, they are not causing the directly immediately death of fish organism, but will cause the long-term effects. So they also accumulate to cause a trophic level and also cause a problem for multi-generations exposures. For microorganisms, actually they also will change. So there's a simple concept drawn graph to show how the microorganisms change under the emerging contaminants exposes. So basically, they may have in original local communities, they will have a lot of sensitive microbial taxa. But under such kind of environmental stresses, the sensitive taxa may decrease and only tolerance taxa can survive. So they finally will cause a change in community composition and functions, and also may cause some microbial risks, because the current study already proves that such kind of microorganisms, even they are non-antibiotic, and also can cause the promotion of spread of antibiotic resistant genes. And so first, if we want to know and to evaluate the effect of emerging contaminants, we need to have a method to determine the compounds, right? So for here in Jolo River, we use the environment chemistry approach and to identify the more than 50 compounds in our waters in North River, West River, and also estuary in different seasons. So finally, we can find 42 compounds. We can identify 42 compounds. So their concentration is not high, right? Nanogram per liter. But we also can find the highest concentration of compound caffeine and bisphenol A. If you drink coffee, if you drink tea, definitely will release caffeine. And if you use the plastic bottles, also may release the bisphenol A. So it's very common. And we also found the antimicrobial agent like triclosan. In our toothpaste, there are such kind of compounds. We also use preservatives to protect our food. And all we use for ladies, they will use sunscreen, right? So they're also common in our rivers. So we want to identify the effect, but we know the pollution concentration is quite low. So how to identify the effect? So here we incorporate the concept of net size. Here is a figure show the social network from Facebook. Yeah, right now we use WhatsApp, we use WeChat. So we will have a social network, right? For microorganisms, they also work together. We also have like different mechanisms to contact each other, to work together, and to maintain the existing functions. And for network, if the community can form network, definitely they will have keystone. Yeah. So that means the keystone will have the disproportionate large effect on its environment, because they have a low abundance. For here network, only keystone, right? They have low abundance, but they are critical. If we remove the keystone, the network will disrupt. It's like a foundation of the building. So for our case, we can identify the keystone species. They are rare. We also can identify the abundance. We can see here the blue bar show the abundance and richness of keystone. We can see here in some part they are quite high in network. That means in whole community levels, the keystone species have very low numbers, abundance, but in network they may play important role. And in network, so we can build a network for river microbes. We can identify the keystone species, and then we can identify whether the emerging contaminant can cause a problem for keystone level. This blue bar shows their effect for keystone, and we also can compare the effect of contaminants to nutrients. So we can see here, yes, they have a strong effect on keystone level. And in addition, they also have a negative effect for biodiversity. We can see here what kind of compounds can cause such problem, like antimicrobial agent like fungicide, antibiotic, stimulate, and preserve, plastic method, and such kind of compounds. So in other words, the emerging contaminants not only resulting in loss of microbiodiversity, they also play as reverse oppose their effect of keystone and may cause the totally change in microbial communities. And their effect actually is larger than the nutrients, the conventional contaminants. And we also trying to identify whether they have a different effect of different living style. We know microorganisms have different life strategy, like for living. They live there like single cells, but they also can live together to attach to a particle to get more nutrient compounds. This work was done by Bob, and we can identify the critical contaminants like antibiotic, caffeine, and something like that. And also we can see from this network, we not only include the microorganism, but we also put the chemicals there, so we can know which chemical pose the effect for certain microorganisms. For this case, we can know that the caffeine have a negative effect on ammonia oxidizing archaea. Why this kind of species are important? Because they are first step of nitrogen cycling. In other words, if no these species, the nitrogen cycling can't work well. So in previous slides, I just show work at the field levels. So we need to prove whether they are true methods. So we did some lab scale experiment. We add bisphenol A and triclosan and the similar compounds at the environment concentrations. So we can see here if the triclosan and if we mix different kind of compounds there, we can see they have different succession weight as the control. That, in other words, some compounds may cause the totally change in microcommunities. So why? This is because if these compounds enter into the surface water, actually they cause the change in the local environment conditions. They pose the pressures to force the microorganism to change. As I said, the tolerance can survive, sensitive disappeared. And they not only will pose an effect for microorganisms in rivers, but may also pose the negative effect on conventional microbiomes of animals. Like here, we investigate the compound effect of tricycline and the microplastic, the compound effect of gut microorganism of typical fish. We can see here if we put those two kind of contaminants together, actually we will lead the different source. I mean, the gut will have a different source of environment source. Gut microorganisms have a different environment source, and also they will cause the change in gut microbiome network. So for the compound, in fact, we can see here is quite clear that they have a simple network structure as compared to control, right? So not only that, they also cause they may lead the pathogens become more important in the network. That means when we put multiple contaminants together and in the river water, actually fish may get sick. So that's a problem. But for gear, there's no clear change. And why? I mean, whether the network really become weak, and we also use those stochastic analysis to show that if we remove the certain species from network, yes, we can see when the microplastic and antibiotic and triclosan together, they have like very weak ability to resist the such changes. Okay, so finally, I would like to use to conclude that, I mean, this summary. So first, in this in this presentation, I showed that high-support molecular technology actually can enhance our understanding of environmental microbes. And the bioinformatic and stochastic approach that could use for data mining, because we know that sequencing generate the big data. And the environmental microbes could be used to identify the pollution sources and to assess ecosystem health. And we also show that if we can incorporate the approach from multiply displaying memory studies at these areas, and actually they could provide a more in-depth insight into environmental issues. And finally, I would like to thank for the support from our team and the cooperators, and also thank for the team support from CAS and the National Science Foundation of China. And okay, finally, thank you, and welcome China. Welcome to Xiamen. Yeah, thank you.
Thank you so much, Prof. Anyihu, for your very insightful presentation. So knowing that the impact of antibiotic resistance is massive, very big, more than diabetes, I think we should have a lot of research regarding this topic in Indonesia, right? So I think as environmental geographer, we can take part in some research part, like the sampling processes, analysis in land use, and etc. So maybe we will continue the question and answer session. So if one of you in this Zoom want to give a question for Prof. Anyihu, you can write it in the chat box, or you can directly open your microphone, and also maybe open your video. Please, is there any one of you? Yeah, maybe we will wait for the participant to give the question in the chat box. And please raise your hand if you want to ask some questions for Prof. Anyihu. And I have a question, Prof. Anyihu.
Yeah.
So actually, what is the best way to reduce the antibiotic resistance in Indonesia? Because mostly we have a kind of the septic tank who will directly flush into the river bodies, and then we also lack of the communal wastewater treatment plant, and also there's a potential of the antibiotic resistance brought by the runoff from the land use to the water bodies.
Okay. So what's your question? Sorry.
So what is the best way to reduce the antibiotic resistance in Indonesia, especially? Yeah.
Okay. So first we need to identify their spread way and their pollution sources. So that's why we did those work to identify the pollution sources in our area. Then we also need to, right now there are some researchers trying to develop some new technology or new materials which can deal with antivirus genes. But I think the best solution is control the pollution sources.
Yep. Yeah, because control the pollution sources maybe is our best practice for reduce antibiotic resistance. So is there any one of you who want to ask some question? Any other question from the participant? Ada. Yeah, okay, there one question from Bapak Ahmad Cahyadi in the chat box. What are the future challenge of this research, especially in developing countries where equipment is limited?
Future solution?
What is the future challenge
Okay.
of this research, especially in developing countries like Indonesia, because we have a limited equipment.
Okay. Yeah. First, for actually, for example, for antibody-resistant genes, and there's a different way to identify the antibody genes. Right now, yes, the high-throughput molecular technology can give a full profile of my antibody-resistant genes because they are huge. They have hundreds or thousands of genes, but not all of them are have a high risk. So actually, the WHO in recent years they release maybe one or two versions to list the high risk antibody-resistant genes. Maybe we can use. I mean, if the equipment is limited, we just can target the certain genes which have a high risk. Another issue is actually antibody genes is the ancient property of microorganism. So that, in other words, this is the natural property. They are natural property. We can't remove all of them. It's impossible. So we need to care of whether their pathogens have antibody-resistant genes. So maybe we can use a different kind of method, just track the evolution of certain pathogens and their spread pathway.
Okay, thank you, Prof. I think I have no question in the chat box. And well, actually, there's some of our colleagues from the biology also join this seminar and webinar. And then I think, yeah, as environmental geographer, I think we also have contribution in examining how the geographical factors influence the distribution and spread of the antibiotic resistant genes in the various ecosystem. And it's also involved the studying of the interaction between the environmental factors such as soil, water, air quality, and land use. And then I think this understanding also helped the researcher identify the hotspots of the antibiotic resistant, tracks its movement and its developed strategies for mitigating impacts in the human health, especially for us in the developing countries, because we still lack of the wastewater treatment plant. And I think that's our, what is it, PR? Challenge, yeah, home, yeah. Thank you so much, Prof, for being with us today. So we will also have a discussion next in the international seminar in our department in September 26. Yeah.
Yeah.
Yeah. Thank you so much for the troubles. And then we hope we can have a lot of collaboration after this. Yeah. Also sure. Invite our college, our friends from the biology and also animal science for this project. So thank you so much. Do you have any last comments for this in this session, for this webinar?
No, and thank you for your question, and so thank you for all audience, and sorry for the delay. Yeah,
it's okay.
Sorry.
It's okay, Prof. So maybe we can have a photo shoot in a Zoom. But Nacho, maybe you can count into, and the host will take the photo. Okay, maybe you can help with the photo session, Nacho.
Okay, thank you everyone for the very enthusiastic and very enthusiastic, and also thank you for the professor and you for the very interesting topic today. And we will have a photo session, so please open your camera. We will wait 1 or 2 minutes, so make sure that everyone open the camera.
Professor Anihu, would you mind to stop sharing your screen so we can see all the participants in the screen? Yes, thank you so much.
Okay, okay, maybe we can wait it. So it's only less than 10 people open the camera. So okay, a few minutes. Okay. Okay, I'll count 1 until 3. So please give a best smile. 1, 2, and 3. Okay, once again, the next slide. 1, 2, 3. Okay, I think this is the last slide. 2, 3. Okay, I think that's all for us. So, okay. So, ladies and gentlemen, we have come to end a very enlightening and informative seminar. And I hope you all have enjoyed and benefit for the presentation and discussion of our distinguished speaker and guests. And I'd like to thank once again for valuable contribution and for making this seminar very successful. And please give a big proud and applause to everyone who has made this possible. I also want to give a reminder, especially for GAL block students, don't forget to fill the presence form in the chat box. And as we conclude this seminar, I'd like to leave you with a quote by Albert Einstein. So the important thing is not to stop questioning. Curiosity has its own reason for existing. I hope this seminar has sparked your curiosity and inspired you to learn more about the topic we have covered today and discussed. I wish you all the best in your future endeavors and hope to see you soon again in our next seminar. Thank you and have a great day. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much, Prof. Anihu. Bye.
Thank you. Bye-bye.
Bye.